Now, when we talk about transport, we tend to focus on the solutes and how solutes like glucose and proteins and ions can travel into and out of a cell. But I want to talk about a specific type of transport called osmosis. Osmosis is diffusion, so it is spontaneous in nature. Okay. However, rather than focusing on the solutes, we're going to focus on the solvent. We're going to focus on the movement of water across a semi-permeable membrane. Now, we talked about the fact that water has a moderate degree of permeability. So that means it has a moderate rate of transport both into and out of a cell. What impacts the direction in which water is going to move? Now, this is an interesting problem because when we talk about the movement of solutes, we often say solutes move from high concentration to low concentration. And concentration is really measured in molarity. Molarity is moles of solute. Per liter of solution, in this case per liter of water. So it's the amount of solute per volume of solvent. Now what's interesting is, how can you have different concentrations of water? Right? Water is water. I can't say this water is too dilute. Can you make it stronger? Right? So we don't really have concentration gradients in those regards. We don't have it in the traditional sense. However, what we're going to see is that the amount of solutes present in the water is going to impact how water moves. So water always is going to flow from an area where there is a low solute concentration. We call this type of solution a hypotonic solution, a very important term, to an area of higher solute concentration. In this case, that's considered hypertonic. Now, rather than just talk about it, let's look at this in a little bit more detail. Here we can see, in the case of a hypertonic solution, we have lots of solute. Okay? So let's imagine in this case, here we have a glucose solution. What we've done here is we've created sort of an artificial system in which we have a membrane that's selectively permeable. What that means is glucose cannot get through the membrane, but yet water can. And now across the membrane we have here a glucose solution within this glass tube. Here we have it bathed in just water. Now you notice here that the glucose molecules, because they are polar, they're going to take up the water molecules. Right? The water molecules are going to form what we call spheres of hydration. So if you look at the water molecules that are unoccupied, there are far fewer unoccupied water molecules here on this side where there's the glucose, or another way to say that, there's much lower amounts of free water in the hypertonic case than compared to the hypotonic case, in which all the water molecules are unoccupied and available. So we say a hypotonic solution has a high free water due to its low solute concentration. And here we say in a hypertonic case, we have low free water due to the high solute concentration. Now, because osmosis is an example of diffusion, in this case specifically the diffusion of water, we see that water moves from where there's high concentrations of free water to where there's lower concentrations of free water. Or another way to say that is water moves from hypotonic to hypertonic. And as a result, what we would see here in this example is the water will begin to rise within the glass tubing due to the fact that water is being sucked in. Water is moving spontaneously down its gradient. And you can measure that distance. Now, when will this transport of water across a membrane reach a net rate of 0? When it reaches equilibrium. When it reaches equilibrium, that means for every water molecule that's going in one direction, there's a water molecule going in an opposite direction. Now remember, when we measure water, we're doing so as a ratio. So when the moles of solute on one side per liter are equal to the moles of solute per liter on the other side of a membrane, that's when we reach equilibrium. Let's see this in actual motion. I want you to pay attention now in this animation. Which side represents the hypotonic side? Which side represents the hypertonic side? The second thing I want you to pay attention to is which side has more free water, which side has less free water.
Imagine a container divided by a membrane that is permeable to water but not to proteins. We can pour water into the left compartment and the same volume of protein-rich solution into the right compartment.
Okay, let's establish ourselves now. We have a semi-permeable membrane. What that means is water can freely move through, but proteins cannot. Which side here, we'll call this side A here. Call this side A. We'll call this side B. Okay? Which side represents the hypertonic side? That's right. B represents the hypertonic side due to the large concentration of solutes that are dissolved in that water. Which side has a higher concentration of free water? That's right. A has a higher concentration of free water. Due to the lack of solutes, these water molecules are moving freely in solution. However, on the hypertonic side, due to the presence of these proteins, some of the water molecules are taken up, forming these spheres of hydration.
As you can see, the right compartment has a lower concentration of water molecules. Proteins take up some of the space. Water molecules can move in both directions, but the net diffusion of water in this example will be from left to right, from the area with the higher water concentration to the area with the lower water concentration.
Now, the things that are important here that I want you really to pay attention to. This is a spontaneous process. Water is going to freely move across a membrane in both directions. Now, you notice he said that. They move in both directions. However, the rates are unequal. The rate of moving water from hypotonic to hypertonic far outweighs the rate of water movement from hyper to hypotonic. So when you look at the net gain and loss, there's going to be far more water molecules gained on the hypertonic side compared to that on the hypotonic side. Now, this is very important when it comes to cells, as you would imagine. Cells are made mostly of water. It's very important that cells are able to maintain a certain volume. So osmosis becomes important to regulate in cells. It also, if you think about it, is going to affect the concentration of chemicals within a cell. If a cell loses too much water, the concentration of those chemicals go up, because remember, concentration is moles per liter. If we lose too much volume, the molarity is going to increase, effectively changing the chemical content of our cell. So it's important as an animal cell or a plant cell, a bacterial cell, it's important that we're able to regulate this net movement of water. Let's see why. All right. Now, let's first establish that cells have a certain concentration of ions in the cytoplasm, has a certain concentration of sugars, amino acids, etc. So cells have solutes. Now, because it has solutes, are cells always going to be hypertonic in nature? The answer is no. Cells are not always hypertonic. The only way to establish if a cell is hypertonic is to examine the environment. So is the environment hypertonic or hypotonic compared to a cell? We have to establish that. Now, cells, specifically animal cells, are interested in maintaining balance between the extracellular and intracellular sides. We want our solutes, ultimately, we want our molarities of our extracellular side, the molarity of the cell, to be equal, and we would consider those two solutions compared to one another isotonic. It's important for an animal cell to maintain water balance. If it loses the ability to maintain that balance, another word for balance is homeostasis, what results is a distortion of a cell's shape. Now let's look at examples. We're going to compare and contrast plant and animal cells. Now let's first establish and reiterate what we learned in Chapter 5. What is one of the major differences between a plant cell and an animal cell? That's right, the presence of a cell wall in plants is very different from the outside of an animal cell. There's no cell wall in an animal cell. That's going to become very important when we start to talk about how a cell can be distorted based on the gain or loss of water. So let's talk first about an animal cell here. Here we have a red blood cell, a nice example of a normal animal cell. You can see here that in a red blood cell, it has a nice circular shape. We see actually that under a microscope, even though this is an artist's rendition, under a microscope, we have this indentation in a normal red blood cell. That's what a normal red blood cell looks like in an isotonic solution. But now, what's going to happen to that red blood cell if you place it in a hypotonic solution? Well, let's think about it. Remember, cells have solutes. I place a cell in a hypotonic solution. Immediately, that establishes the cell then as being hypertonic. And if the solution is hypotonic, which way is water going to move? Into or out of a cell? Well, as we've established, water always moves from hypotonic to hypertonic. So the cell's going to begin to swell. You can see it's already distorted. We see the loss of that nice rounded shape. We see that it's more oval and not as uniform. We see the loss of that indentation. So now we've seen a swollen cell. If the cell can't control the intake of water, eventually that animal cell is going to be ruptured. It's going to lyse. We can see that here. That's cell lysis. And what's going to happen as a result is the inner contents of the cell are going to spill out, and that's going to result in a dead cell. Now let's look at the converse of that. Let's look at if we place a red blood cell in a hypertonic solution. Because of that, that establishes my cell, in this case being hypotonic. And because the cell is hypotonic, we are going to lose water to the environment. What happens again is we distort the cellular shape. The cell is going to shrink, or plasmolyze as they call it. Which is also bad for a cell. When a cell loses water and shrinks, what's going to happen is the volume decreases. We need a certain volume in the cell in order to carry out metabolism. We are also going to see an increase in the concentrations of certain solutes, which is going to affect the balance within a cell. In either case, whether it's the cell shrinking or the cell expanding, both of those are bad for for animal cells. Therefore, animal cells work hard to create an isotonic environment. What organ really is involved in regulating this isotonic solution? If you think about the human body, the kidneys help maintain this isotonic environment, because kidneys can either release or absorb water, and they can also release and absorb different ions, which is important as well. So if we have a hypertonic environment, what the cell is going to want to try to do is retain more water to try to dilute it and excrete more salt. So now if we have a hypotonic environment, what we're going to try to do in this case is we want to retain salt and we want to get rid of excess water. So the kidneys are either excreting or are absorbing water, excreting or absorbing salts in order to maintain this isotonic environment. Now, a plant cell is different based on that cell wall. Right? So now let's see how a plant takes care of water balance. Now let's imagine in this case, I place a plant cell in a hypertonic solution. Because the solution's hypertonic, that must mean the cell is hypotonic. Therefore, the plant cell is going to lose water. And as a result, the volume inside the plasma membrane shrinks, and the plasma membrane starts to pull away. You can see that here, starts to pull away from the cell wall. Again, plasmolysis, not good for animal cells. Plasmolysis, not good for, crenation, not good for plant cells. Now, in the case of here, in a hypotonic solution, that means that a plant cell is hypertonic, and as a result, water is going to enter the cell, and what we see is the cytoplasm starts to expand. Right? However, because of the cell wall, there's only so much room for that cytoplasm to expand before the wall starts to push back. We're going to call this turgor pressure. And what we'll see here in a minute is that a plant cell is going to prefer a hypotonic solution because that's going to allow a plant to grow and sit up against gravity. So we can see already the difference between a plant and animal cell is the solution it prefers to be in. And also, as we talked about, the presence of a cell wall is going to prevent major expansion, where the absence of a cell wall may ultimately result in rupturing of an animal cell. Now let's look at this example. You can actually apply this to both plant and animal cells. And I want us to walk through and think about what's going to happen in each case. So here we have a dialysis bag that's filled with 98% water, 2% sucrose. Now, consider this to be a cell. And remember, a cell has a semi-permeable membrane. In these bags, the bags are permeable to water and not sugar. So the only thing that we're going to regulate the movement of in this case is the movement of that water. Now let's imagine we take this cell, 2% sucrose, 98% water, and we compare it in the first scenario. Let's say the environment in this scenario is 100% water. It is hypotonic. That means there are no solutes. If I place this bag into this beaker, what's going to happen to the size of that bag? Well, let's take a look. Let's take our cell here, our artificial cell with 2% sucrose. Let's drop it into our hypotonic environment. In this case, the cell is going to expand. Now, why does the cell expand as I put it into a hypotonic environment? The reason is simple, because if you look inside the bag, we have higher amounts of solute. The bag in this case is hypertonic compared to the environment. And remember, water will always move from hypotonic to hypertonic. In this case, water is moving from the beaker into the bag. Now let's look at the second scenario. In the second scenario, we've changed the environment. Now we have a hypertonic scenario in which there's 10% sucrose. Now the first question I'm going to ask you is this. What has more free water? The artificial cell or this hypertonic solution? That's right. The cell has more free water. There's only 2% sucrose. That means that the rest of the water is going to be free in solution. When you have 10% sucrose, many more of those water molecules are taken up by that polar sugar. So there's much more free water in the bag compared to the beaker. Now, what's going to happen to the size of our cell, or the shape of our cell, if we place it in that hypertonic environment? Well, let's take a look. You notice what happens to the bag? The bag deflated. Now, why did the bag deflate? Because as we just established, there's more free water in the artificial cell. Therefore, the water is going to move out into the hypertonic environment, causing that cell to shrink. So we see the difference here. In a hypotonic environment, the cell expands because water moves into the cell. In the opposite case, in a hypertonic environment, the cell shrinks because the water is going to move out into the environment. So now let's look at the third scenario. Let's look at the isotonic scenario. What has more free water? The artificial cell or the solution in the beaker? That's a trick question, right? They have equal amounts of free water because they have equal amounts of solute. So what's going to happen to the shape of this bag or this artificial cell as I put it into the third beaker, which is isotonic? Well, let's see. Nothing happens to the shape of the cell. Why? Because the rate of water movement out of the bag is the same as the rate of water movement into the bag. Therefore, at an isotonic state, we have equal rates of water movement. And what we establish in this scenario is that water balance has been established. This is the environment an animal cell prefers to be in. However, what's the environment a plant cell prefers to be in? That's right, a hypotonic environment. Now, if this is a plant cell, let's say this is an artificial plant cell and we put it in, do we worry about the cell bursting? Do we worry about this bag bursting if this is a plant cell? Why not? Why don't we worry about it bursting? That's right. Due to the presence of a cell wall, that plant cell is going to fill with water until the pressure of the wall on the cytoplasm is equivalent, until we reach equilibrium. Now we can see why this is important for a plant cell. If you've ever not watered your plant for a few weeks, you notice what happens, right? It starts to droop. And then all of a sudden, when you water it again, what happens? It starts to stand up tall against gravity. What's the difference? The difference is simple. In this case, when you don't water it for a while, the soil becomes hypertonic. Therefore, water is drawn out of the plant, causing a loss of trigger pressure. But now when you water that plant, you're now creating a hypotonic environment. And because the soil now is hypotonic, water is drawn into the plant. And what that's going to do is reestablish the trigger pressure required for a plant to sit upright against gravity. Right? We sit upright against gravity because we have a skeleton, right? But plants don't have a skeleton. So how can it maintain this upright position? It all has to do with the flow of water into and out of a plant. Now, as we talked about, water has moderate permeability. And for a long time, it was just thought that water, even though it doesn't move at a high rate, can simply just move into and out of a cell. However, that doesn't make sense for organs like kidney cells. For cells like kidney cells. Kidney cells have to be able to rapidly either excrete or uptake water. How can a kidney cell function with this moderately permeable water? Well, they've established that in cells like kidney cells, cells of the bladder, red blood cells, any cells that have a need to regulate water balance, that there's actually proteins called aquaporins. These aquaporins are transmembrane proteins, and they simply just allow water and environment where they can move quickly into and out of a cell. This was a major breakthrough. I think this was established probably somewhere around 1995 and 1996. Here's the three-dimensional structure of an aquaporin. You see the presence of all these alpha helices that insert themselves into a membrane, and because of these alpha helices, typically there's 7, we create this barrel-like structure which creates a channel in order for these water molecules to move. Now this is actually an interesting case study in science. How did they know these aquaporins were present? How did they discover them? I'd like to just quickly walk you through how they did it. And I think this is really interesting to look at the scientific process. Okay, so now here's what they did. They first, at the experimental level, what they wanted to do is they wanted to make copies of a gene. So they started with this hypothesis that there was a gene called CHIP28. And based on some anecdotal data or some preliminary data, they had thoughts that this actually acted as a water channel. So the protein produced by the gene CHIP28 functioned as a water channel. Now, in order to carry out this work, they did a little bit more digging. And what they established was that red blood cells and kidney cells had a lot of this CHIP28 protein. Now, that was interesting to them. Why would kidney cells and red blood cells, as compared to others, have lots of this protein? Well, they looked at the function of these cells. And they certainly know that kidney cells have an important function in maintaining water, either by excreting it or retaining it. They also know that red blood cells, it's important that we maintain water balance in red blood cells so they don't burst. So now we have a protein found in cells involved in water balance. Possibly CHIP28 was involved as a water channel. So what they needed to do is they needed to establish a cell that lacked CHIP28, lacked this CHIP28 protein. And their goal was to insert the protein and then look at water uptake into that cell that contained CHIP28. So how in the world can you insert a protein into a cell that lacks the gene for that protein? Remember, we learned about this in chapter 3. DNA is transcribed into RNA, which is transcribed into protein. How can a cell that lacks the gene, the DNA for CHIP28, ultimately produce the protein? What they did is they produced mRNA in a test tube. They included the enzymes important to produce RNA, called RNA polymerase. So what they did is they added the nucleotides necessary to make RNA, they added the enzymes, and they added copies of the CHIP28 gene. So they put the DNA in, they put the nucleotides in the test tube, put the enzymes, and as a result, they got lots of copies of the RNA for that particular gene. Now recall, the process of converting mRNA to protein is carried out by what structures? That's right, ribosomes. Well, all cells have ribosomes, so once I put the RNA corresponding to CHIP28 into a cell, the ribosomes for that cell should produce CHIP28 protein. And the cells which they used to do this were actually frog oocytes, essentially frog eggs. And using very fine needles, what they do is they puncture the plasma membrane without destroying it, and into the cytoplasm they introduce the CHIP28 mRNA. Now, what they did then is they waited several hours. They allowed the ribosomes to begin the translation process. Once that protein was translated, that protein was ultimately inserted into the membranes of oocytes. So now, let's set this up into two groups. We have a normal oocyte that lacks CHIP28 Then we have the experimental oocyte. While it doesn't have the CHIP28 gene, it does produce CHIP28 protein. So now in a normal oocyte, the plasma membrane lacks these putative water channels. In the experimental oocyte, we have these series of proteins who we hypothesize to function in water transport. Recall, at this point, they have no idea what the protein does. They've hypothesized that it's involved as a water channel. So now let's think about this. If I take two different beakers, we'll call this the normal beaker for the normal oocyte. This is the beaker for the experimental oocyte. And let's say we have a hypotonic environment in each scenario. And we put these cells in. Theoretically, right? If this protein functions as a water channel, what's going to happen to the shape of the experimental cell compared to the shape of the normal cell? What would you expect based on everything that we've talked about? If CHIP28 functions as a water channel, I would expect the cell in a hypotonic environment to expand rapidly, whereas I would not expect there to be much growth in the size of these cells because the rate of water movement is much slower due to the absence of these water channels. So from a hypothesis standpoint, that's what we would expect. Well, did the experimenters see that? Well, what they did is they put, as we talked about, the oocytes into a hypotonic medium. Okay? They had their control group that did not have CHIP28. They then looked at the cells and the shapes of the cells under a microscope. And what they saw is exactly what we would expect. Here's what the cells look like before treatment. We can see that each of the cell has a characteristic volume that's quite similar. Now, when we put it into a hypotonic environment, each cell, we notice there's no change in the control cell that lacks CHIP28. However, now in the cells that have this protein CHIP28 in their membrane, we notice a pretty stark increase in size. Actually, such an increase in size that we can see here the integrity of the plasma membrane has been affected. And now we can see the oocyte begins to rupture. So now they've repeated the experiment multiple times. In every case, they see this cell bursts while the control cell remains the same. Do these results support the hypothesis that CHIP28 acts as a water channel? It's clear that the rate of water uptake is much greater in the presence of this protein CHIP28 compared to that of a cell that lacks CHIP28. So based on these results, instead of calling it CHIP28, they renamed them to call them aquaporins. And it's clear, based on these results, that aquaporins function to increase the rate of water uptake, which is extremely important in cells like red blood cells and kidney cells.